Good morning, everyone. I can just see you're all coming in to the showcase. So good morning and welcome to another spring showcase. Welcome from Cardiff University this time. So we are with Odour and Cardiff University for this eDNA taste and odour monitoring showcase. So for anyone who doesn't know me, Carly Perry from Spring Innovation. We are the Innovation Centre of Excellence for the water sector, supported by all of the water companies across the UK and Ireland, and with the goal of removing duplication and accelerating innovation in the water sector. So we're doing that through a couple of service offerings. One is the Spring Accelerator, where people can submit ideas into us and we'll build collaborative projects to remove duplication. And the second is this knowledge transfer service offering that you're going to be experiencing today. So there's two core pieces to that service offering. One is a knowledge library on the Spring platform. So you can go there and access case studies, learn from insights that others have had in the sector. And these showcases, so these are 2-hour deep dives that allow you to learn from project teams who've been there, experienced something, had the challenges, and can now share the insights that come out the back of that. So what you'll be learning today is quite a bit of technical information about this project. Bear with me whilst just figuring clicker pieces out. Clicker is not working, so bear with me 2 seconds. I'm just gonna. No, this is not working. Sorry, everybody, just bear with us whilst we figure this out. I'm going to use there we go. It's now come back to life. There we go. Sorry about that. Okay, so this is going to be a 2-hour deep dive into some insights from the project between Welsh Water and Cardiff University. We've got a great group of people for you to hear from this morning. So in a second, you'll be hearing from Tony Harrington over video. He is actually on leave at the moment, but he wanted to be able to share his thoughts with you. You'll be hearing from a team at Cardiff University, as well as Paul Gaskin from Welsh Water. So ultimately, these knowledge showcases are all about you engaging with the knowledge. So importantly, we want you to learn the lessons and take them away and embed them in your day-to-day jobs. So at the end of this, we'll be doing a poll to find out how interesting you found the information and whether you're going to use it afterwards. So we'll be following up with anybody who's going to do that. So our KPI is all around making sure the knowledge from these sessions is embedded. So your feedback is really helpful for us to do that. So with all of that in mind, I am going to hand over to Tony Harrington on video.
Hello, everyone. My name is Tony Harrington. I'm Director of Environment at Welsh Water. I want to welcome everybody to today's knowledge sharing showcase. I hope it goes well. I'm really sorry I can't be with you. I'm actually on holiday in Austria as you're sitting there. But anyway, as part of my role at I look after innovation. And we also work with a whole host of partners, many of whom are here today. One of those partners is Cardiff University. We have a strategic memorandum of understanding with Cardiff and have had very close working relationships with Cardiff and several other universities, actually, like Bangor, over the years. So particularly welcome the academics that are here today. We look forward to working ever closer, really, with you and the whole university sector as time goes on. I wanted to just say a little bit about why knowledge sharing is important. A few years ago in the water industry, we tended to do our innovation and research work, not so much the research, but mainly the innovation work in miniature silos, really, that were either siloed according to skill and practice, so on the water side and the wastewater side and so on, or alternatively, they were done almost secretly within water organisations to get competitive advantage over sister organisations elsewhere. That's now changed, thankfully, and we do much more of our innovation work together. collaboratively, and that's led by Spring. Spring is the centre of excellence for innovation in our sector, both for the UK and Ireland, and long may it continue. The efficiencies that we can gain from sharing our knowledge and thereby the benefits to customers and the environment that will be accrued from that effort will be considerable. We very much support these types of sharing events, and will be participating in future sharing events. We very much look forward to that. So without further ado, I'll pass you on to colleagues now, and I hope you have a really good session. Thank you.
Wonderful. So that's Tony Harrington, Director of Environment at Welsh Water. What we're going to do now is get into the programme of activities. So I'm going to hand straight over to Rupert from Cardiff University.
Hello everybody, and thank you very much for turning up, and welcome on behalf of the Cardiff team to Cardiff University. So as many of you probably know of me as Rupert Perkins, I've been working with the water industry closely now since 2016, and primarily connected with Taste and Odour. One thing I need to point out is if you have questions during the presentation, my part and then Sophie that I'll be handing over to, and then Pete after that. If you have any questions, please do feel free to put them in the chat. So what I'm going to do is start by giving you a brief sort of background to the project and how it started going forward. Then I'll be handing over to Sophie, who'll be giving you far more details about the methodologies and the knowledge transfer, and then over to Peter Kille, who'll be talking to you about and giving you a demonstration about the kit so you can see it for real. So what are we talking about? Reservoir community analysis for evidence-based management. So this is the title of the project that we put into Ofwat. So my role in this was working with the water industry partners, who are named at the bottom, many thanks to them, obviously, in designing the application that went into Ofwat. And as the ecologist on the project, I'm the person who sort of tries to take the eDNA data that we get and make it into a useful, active tool that can be used then in the water management. So many thanks to the water industry partners. There's a whole list of names there as well that I'd like to draw attention to in brackets. This is a big project. It's involved lots of additional value added from various other projects as well. So many thanks to Annalise, Amy, Inga, Veronica, Charlotte and Tom as well, and others that I probably should be mentioning too. So as I said, I'm going to give a quick background, then we're going to have Sophie talking about the molecular approach, then the technology demonstration from Pete, then we'll be having the quick break, and then afterwards what we've learned and the way forward before our question and answer session. So starting off, what's the problem we were actually focused on? Although eDNA can be used for many different problems and applications, we were primarily focused on taste and odour to demonstrate its capacity. So quick background, but I'm sure most of you, if not all of you, already know about the taste and odour issue. We're talking about metabolites geosmin and 2-methyl-isoborneol, which are produced in reservoirs, typically cyanobacteria. And they produce this earthy and musty taste and odour that leads to customer complaints. So the primary source then are filamentous cyanobacteria. But what we were trying to demonstrate was how we could embed this technology into understanding the triggers to facilitate intervention solutions, or preferably prevention and also optimization of treatment. So the problems behind the problem. Okay, so when we're studying taste and odour, we need to bear in mind this is not a eutrophication problem. It's not the classic elevated phosphate type of story that many people associate, for example, with algal blooms. And it's also not an algal bloom related problem. It's not cyanobacterial biomass we need to be worried about. It's a combination of nutrient triggers, environmental variables, biological processes, which makes it a complete nightmare. Because all of those different factors, all those variables interact together, which makes it very difficult to actually determine what specifically is causing the problem at a specific site. So what we wanted to do was turn the problem on its head. Basically use the cyanobacteria and the community of algae as bioindicators of the risk to determine the triggers and hence enable prevention and also prediction of taste and odour events. So by the community, what we're talking about then is not just the cyanobacterial producers, but the diatoms that they have intimate relationships with that ultimately lead to them having nutrient availability, green algae, and also bacteria. And it's a functional analysis because some of these bacteria are crucial as well within the process. So can we combine with water metadata, such as nutrients, DO conductivity, the eDNA data to actually generate what's useful to water industry, a traffic lights approach, basically, red, high risk, amber, moderate risk, green, everything's okay. So what's the problem behind the problem behind the problem? Well, that's because cyanobacteria are sneaky little things, basically. The identification of algae and cyanobacteria isn't easy. And if you've ever looked down a microscope, you'll realize that looking at cyanobacteria is particularly hard. So when you're identifying these species, if you go into genus, you're doing well. Unfortunately, if you go into genus, that is not sufficient in being able to predict taste and odor or understand taste and odor events, because genus can be a whole range of species, and within those, some will be producers and some aren't. And we know from looking at studies like publications on the identification of chorobium and other species that even when you think you got it to genus, you could still be wrong. So the accuracy of that data is difficult. If you're going as far as species because you're an expert and there's not many of those left around, it's very, very hard, and it can still be insufficient because what we found is that strains of the same species can be producers or not producers. So ultimately, we need to identify the actual key producers as the ultimate goal, bearing in mind that not all strains of the same species are the producers. This is made even worse because 2-methyl isoprene is produced largely by benthic species, the species that live on the periphery and the edges and the sediment bottom of your reservoirs. And we're not going to detect those typically in normal water samples and then looking at them with microscopy. So we've got a problem that we basically need to try and find a way of looking at this community and identifying the species and then looking at how they interact and use those to tell us what's going on in the reservoir. So that leads me on then to the solution in detail. So what we're actually driving towards then is a near real-time, accurate eDNA community data analysis data set for reservoir monitoring. What is viable, what is practical for people to do in the field to facilitate fast, responsive management. So we're trying to be predictive in our management and ideally preventative in our management. And this is where the eDNA technique comes in a really user-friendly manner. So I'm going to hand over now to Sophie, who is the postdoctoral researcher on the project and the person who's doing all the real hard work behind it. Okay, over to you, Sophie.
Hi everyone, thank you really for that introduction. I'm just going to go through some of the methods and hopefully show you how easy it is to implement this kind of stuff into your own laboratories. So as Bruce just said, complaints related to taste and odour cost industry a huge amount of money each year. And the issues are only likely to get worse and worse due to various climatic and agricultural pressures. The current practices that we do have in place for monitoring taste and odour often aren't timely enough and don't turn around high enough resolution data in order to be able to make management decisions. And unfortunately, for that reason, management decisions are often either delayed, and we're working in this kind of responsive way versus a proactive approach, which is what we're aiming for. And for that reason, there really is this demand for rapid, accurate species level community data. And what I mean by that is we want to know who's there. So we want to know which bacterial and algal communities are dominating those waterways. We want to know whether there's a healthy diversity, what the numbers of producers versus TNO degraders looks like, and whether there's any other kind of water quality indicators that could give us some information. But we also want to know what's happening. So we want to know if there are changes in the presence and absence of those species, maybe the abundance of those species, but certainly the distribution of those species along the waterways. So for anyone who doesn't know, eDNA, it's actually quite a simple concept. So as an organism moves through its environment, it will leave traces of itself everywhere it goes. So that could be dead skin cells, mucus, feces, or free DNA that's already escaped the cells. And you can detect that DNA in pretty much any substrate, so water, soil, even air. So what we can do is we can extract that DNA from any of those substrates and we can target the taxa that we're particularly interested in. So we can design or find pre-made primers that target bacteria and algal communities and then provide the species level information that we need. So you don't have to understand this in too much detail, but this is just a visual representation of the 16S rRNA gene. It's ubiquitous across prokaryotes. And this is just to kind of demonstrate that there are 9 hypervariable regions across this gene. So there are peaks here that you can see, which are really highly conserved regions. And then you can see the troughs here represent really hypervariable information. So if we were designing primers or looking for primers that were published, what we want are primers that are kind of targeted right on that very edge of the conserved region, but flag an area that is very, very hypervariable and therefore gives you all that species level information. And this works in literally the exact same way as reading a barcode in a shop. So in a shop, a barcode represents just a string of binary code, and the order and number of those ones and zeros are in a reference list, and that reference list will tell you if it's a banana or a bottle of water or whatever it might be. And in the exact same way, the string of base pairs that we're reading will correspond to a reference list that will tell us what species they are. So there are different types of technology that we can use to look into this. I'm going to kind of go briefly into 2 of them here. So we're a big advocate for nanopore technology, which Pete will show you a little bit later. But there are other ways of kind of scaling it up. But ultimately, the steps are very, very similar no matter what you're doing. So this is just kind of a shell of what you have to do. So it all starts with your sample collection. So sample collection for us, we use a Smith-Root eDNA filtration unit. Pete will show you this later on and show you how it works. The reason we use it is just because it's very, very simple and we can use it in the field. We team it up with Smith-Root self-preserving filters. You pump the water that you want across the flat filter and it will preserve the DNA on the surface. You crack the filter a little bit and desiccate it, and it means that we can keep these filters for up to 6 months at room temperature. So it's just easy for us for storage. But there are lots of other ways of doing this, you know, around costs or timeframes or whatever. But at this point, this is where you'd want to consider whether you're sampling from the shore directly or whether you're doing boat sampling. You will get very different communities depending on where you were sampling from. But it's also where you'd want to consider the number of sites that you're interested in, the number of replicates you might be interested in, and the number of controls that you might be collecting. So the next step is to extract the DNA from those flat filters or from the type of filter that you've got. There are loads of different ways of doing this. There are commercially available kits, but there's also ways of adapting or optimizing those kits based on what you're interested in. So if you want to optimize by time or by cost, it's possible to do. The principles of it are really, really similar. So there's kind of 5 basic steps. The first step would be to lyse or disrupt the cells to free the DNA. The next step would be to remove any inhibitors from within that sample that might affect your downstream sequencing. You then need to bind that DNA to a surface, so that could be beads, or as this picture kind of depicts here, something that's really commonly available and is cheap are these spin columns, and they've got a membrane on them that the DNA binds to. You then put them in a centrifuge and spin the liquid through, and everything binds to that surface. You then wash the DNA to remove any impurities, and you then elute the DNA, which means you just wash the DNA from that surface so you have sterile liquid at the end of it. The next step that you do is called barcoding. It's also sometimes referred to as indexing. And all that means is you're annealing a known string of base pairs onto the end of the DNA, and it will be different per sample. So you'll use a different barcode for sample 1 versus sample 2 versus sample 3. So you know where those DNA strands have come from. The next thing that you do is a clean. So we use spry beads for this. They're just a type of bead with a magnetic coating, and the DNA binds to these beads. And then anything that is kind of short fragments or impurities will remain within the liquid. You put them up against a magnet and they all move to the side. And then you remove all the liquid that includes all the impurities. You then just do another a quality control check, and you're ready for loading that sequencer after you've pulled them in equimolar concentrations, and then you go on to data processing. So for anyone who doesn't know how a nanopore works, the principle of it is quite simple. It basically monitors the change in electrical current as those nucleic acids are fed through a protein pore. So the kits that it comes with have an enzyme that feeds it through that pore, and as each base pair goes through, it will emit slightly different kind of frequency and intensity of currency. So you will know that what's being fed through is an A or a G or whatever. And the string of those base pairs that goes through will correspond with a reference list, which will give you that species level ID. It's gone a bit funny again. So there is another way to do this, as I've kind of explained. So that's Illumina technology is another alternative that we use. The steps are very, very similar between the two technologies, but this is a way of kind of upscaling what you're doing. So if we were working with lots and lots of samples, maybe in a 96 well format, we might choose to use Illumina so that we can kind of counteract some of those costs. The first two steps are exactly the same, your sample collection and your DNA extraction. But then what you would do is a first round of amplification, which would amplify what you're interested in, so your bacteria or algae or whatever it might be. You then do a quality control check. And then you do your secondary PCR, which would kneel on those indexes, also known as barcodes, if you remember, as well as some adapters, which is literally just something that enables the technology to know it's in the right format to go through. You then again need to pull those samples into equimolar concentration. So we're looking for similar levels of DNA concentration across those samples so that there's less bias on the sequencer. If it's in the 96 well format, we use something called a sequel plate, which is basically a plate where the wells have a known surface area around them. You saturate them with your DNA, it will bind to the outside, all the excess is removed, and then that means very, very quickly you can get them all to a similar DNA concentration. And then you go ahead and you load the sequencer and you go ahead with your data processing. So there's lots of kind of differences between these two technologies. Pete's going to show you the nanopore a little bit later on, I believe. But they're different, but they're both useful in different ways. So the nanopore, the reason that we're really interested in it is it's absolutely tiny. Pete will show it to you. It's about the size of a pencil case. The flow cell is about the size of your thumb, and it only costs about 4.5K. But the Illumina, which you can see in the left-hand picture here, is a little bit bigger. You'd probably need a reinforced bench top for it and a bit of a cooling system. It's a bit more expensive at a 60K. But those costs kind of counteract each other depending on how much you want to scale up. So the technology that you maybe decide to go for would probably depend on the number of samples and how often you're running those samples through your process. And we can leave this slide for people who want to look into the variations in costs and data outputs and whatnot. But the other types of lab equipment you need, depending on how your finance system works, are relatively affordable. A lot of these you can actually get secondhand now if your finance system allows it. And they vary in price. You can really spend what you want to on these. But things like a vortex, a mini centrifuge, and a set of micropipettes are things that people often have in their labs anyway, but they're relatively affordable. And then things like a bench centrifuge, a qubit, which is a machine that reads DNA concentrations for you, and a PCR machine are a little bit more expensive, but you can kind of spend what you want to on those. The other considerations that you might want to kind of play along with are how you process those samples. So you want to be able to keep a sterile environment. You want to remove as much contamination as possible. So some laboratories have the luxury of having separate rooms for being able to do this, but essentially you want to be able to move your samples in one direction. So you'd want to preferably extract your DNA in one room, do your PCR setup in another room, PCR application. You'd never move the samples backwards to prevent contamination. Obviously, not everyone has the luxury of having separate spaces for that. But what a lot of people do instead is they have separate bench locations or different bench spaces throughout the same lab, and it moves in one direction and doesn't go backwards. The only other thing that you'd probably want to consider is freezer space for storing all your samples and making sure that you can keep those samples for as long as you want. -20 is fine, but for long-term storage you might want -80. because once you've got that DNA, there are tons of other applications that you can use with these. So once you've got the DNA there, you can use different primer sets to look at different taxa of interest. So you might be interested in fish, or birds, or invertebrates, or fungi, which means that you could look at fish stocks within your reservoir, or invasive species, or protected species. And that means you can kind of build the picture that you're interested in. You could get a really holistic view of who's doing what, where, and when within your reservoir, which means if you're interested in kind of taking a nature-based approach, you might want to be implementing some of these methods. And that means that you can get a really clear picture of indicators of change within your reservoir. So once you get the data out, there's a various kind of bioinformatic process that you go through. We've got tons of scripts and things if people are interested in looking at those. But ultimately, what you end up with is just an Excel spreadsheet. That in its truest form, that's what you kind of end up with. So if you want to, you can just have a quick and dirty look in Excel and look at your data. But if you wanted to export that and run it in the statistical program such as R, it means that you can model that data alongside other variables of interest, so nutrients or temperature or depth or whatever it might be. But something that we've been doing is we've been converting the data into dashboards. So these are easy to interpret and they're interactive. We've been using a platform called Tableau. There are lots available, but Tableau is the platform that Welsh Water used for presenting their COVID data to the public during the pandemic. So if I could just give an example, this is all just example data. It just kind of gives an idea of the types of things you can put up. You can put up anything you want, but it means that you can very clearly see the trends throughout your data over time. You can click on different points and get extra information. You can exclude points interactively. You can really interact with that data in a really easy to use way. And again, this is just example data, but it means that you can create maps. You can have color coding systems where you're, for example, looking at your counts of your jasmine producers by site, and you can color code it to have kind of a traffic light system of what's going on within your reservoirs. So just to kind of conclude, the eDNA is a really useful monitoring system and can be a really useful early indicator system for management. And it provides such a huge amount of information on where and when you could treat. Importantly, you need to be able to build up a picture of what your norm is. It will really help you get an eye in for when you're seeing big changes within your data. And aligning that data with other variables of interest is really, really important. I will go into that a little bit more later on when we talk about the things that we've learned throughout this this project. So what I'm going to do now is I'm going to pass you over to Pete Kille. Pete's going to go through some extra stuff around the technology and also give you a little bit of a demo.
Thanks, Sophie. Good morning, everyone. And I'm Professor Peter Kille from Cardiff University, and actually I'm a genomics expert. So the kind of tools that this project's exploiting to understand a little bit about the biology of what's going on are sort of my area of expertise. But when we started this project, the important thing for me was to ensure that those tools could be used by the industry. And actually, the partnership with Rupert Perkins, with Sophie's assistance, has allowed us to become real, to basically take the technologies that were research tools and really try and develop something that was robust, that would be useful within that industrial setting. And without, of course, the support of Welsh Water, we wouldn't have been able to do that technology development. So thank you very much to Paul and the people at Welsh Water for working with us. So what I've been asked to do is give you a little bit of an introduction of the technologies. And again, firstly, the technology we want to do is sample the water. And for ages, this was done by taking just a bottled sample water. And in fact, some of our partners have been instrumental in doing this. So we had people from Yorkshire, taking bottled samples, keeping them cool and sending them by courier to Cardiff. Now, obviously, that is not a tractable solution when you're taking samples from across the country. It's not only expensive, but it's highly challenging. So actually, it wasn't going to be something that would be practical long term. So I started looking around for a solution. We initially were using filters that had to be preserved on site. We'll come back to that in a second when we think about developments. But the difficulty with those is, again, you had the issue if you made the filter and you had to keep it cool, or you had to add some chemical preservative before it could come back to the laboratory. Again, something that really is challenging, one, for people that are doing the sampling, but also for that transportation and those storage issues. So actually, we identified some innovations by an American company that has made our life a lot simpler. And these are these eDNA filtering setups and the filtration apparatus. And I'm going to show you that now. What I would say is we started off using a deluxe, a Rolls-Royce version, which I will now show to you, which are these backpacks. Now, you can hear me grunting. So these are fantastic apparatus, magnificent. It has an onboard GPS. It monitors flow rates. It's actually essential if you're, for instance, walking up a stream and you want to detect the eDNA across a stream. But for everyday sampling, it's very heavy, very cumbersome, and actually takes a fair amount of knowledge to operate. So although fantastic and provides a great solution, wasn't again practical for us to use all the time. So again, a great thing, and again, fit for purpose for different applications. The other thing I'll show you is I'm going to show you a filtering apparatus. Now, often when you're going up to a reservoir, the water is not where you'd like it to be. At the moment, the waters are receding. We've got a lovely summer coming, which for everyone means a good playtime, but for the reservoir means they're shrinking. And actually getting at the water can be challenging. So again, one of the other pieces of auxiliary apparatus that we do have is this extendable rod. Now, I'm not going to extend this to show you how large my rod is, but I can extend it to 3 meters if I'd like. So this allows you to actually sample the reservoir water at distance, even if you have a significant mud in front of you. Now, the actual apparatus that we do use, I'm just going to bring on a trolley now and hopefully not destroy the electronics. So this is one. It's actually, they advertised as a citizen science setup, and it's very portable. It has a battery that can be taken out and charged remotely, and actually you can swap out batteries. We use it, we can do up to 20 samples without changing the battery, so it's very practical for a day's sampling. It also doesn't need any skill to use. So again, it's been supplied through the water industry to partners that just do sampling for them, that don't have any biological training. So again, a really practical piece of apparatus. Now, actually, I'm going to put on some PPE. Now, this PPE isn't to protect me from the apparatus, it's actually to protect the apparatus from me. Because what we want to do is we want to sample water, but we don't want to sample a whole lot of human DNA. So when I put these gloves on, I'm not protecting myself from anything toxic, I'm protecting the sample from my DNA so that we're not having a huge amount of human DNA. All the filter apparatus comes in these pre-sealed and pre-sterilized apparatus. And again, nice thing so you can write all the details. Again, we can open up. So I'm going to open this up. I tear off the tab and I remove the filter, and I ensure that I do not touch the end that's going to be used for filtering, so the pre-filter end. I then put a sterile tube that's in the packet, and I put it in while it's in the packet. So again, I'm not introducing any DNA to the front end. I can then put it onto my filtration, and I'm ready to filter. Now, at this point, I need assistance. Paul, my glamorous assistant, will hold the water up so you can see it. And I'll hopefully not leave it there for too long. So I'm going to switch the pump on. And what I wanted to show you is just how rapid this system is. So again, we're pulling water through the pump. And again, what you can't see, I'm going to hold it up so you can see, is the water coming through the pump. Oh, sorry. I've now created an air bubble that will probably take a little time to go through. So here we go, the water's coming through. And again, you can detect the amount of water coming through. That was the air bubble I introduced. Here we go. nice long pipe. And it's much more rapid when you don't have someone introduce an air bubble through. Or how you could select for so it's now dealing with the air that I introduced into the system, which doesn't help. Well, we'll leave it. Oh, it's coming through. So it usually takes about 5 minutes to sample to make a sample. The one thing about the pipe, I'm not going to holding up about 4 litres. Here it comes through. The air's gone now. So this we'll put it down, I think, because 4 litres of water. Let's put it down. Yep. Switch off. So it will pull through about 500 mils in about 5 minutes. You can get about 2 litres through if you have 10 minutes to spare. But what it allows you to do at the bench side is to basically create a filtered water. And as Sophie says, at the end we pull through air and that dries the filter out, and that dried filter can be packed up into the sealed package that I showed you initially, and then can be sent through the post, totally stable for about 6 months. So again, what that allowed us to do is to basically ensure the beginning of the pipe was stable and usable for the industry. Once you've got that, you of course go through the process that Sophie has introduced us to, which is the barcoding and the molecular biology, and then we count the organismal content. And that can be used for a whole suite of applications, from source tracking to anti-AMR detection, through to pollution events, through to water quality analysis. Again, if you also have fish stocks within your reservoirs, you can monitor fish stocks and you can look at invasive species. So actually, once you've got that DNA, there's a huge amount you can do with it. And also, once you've made the DNA, it will store for years. So you can create an archive that you can delve back into again and again and again. So as well as measuring the composition by species, you can also do some quantitative analysis. So we amplify 16S for bacterial analysis, but we also quantitate 16S as a surrogate for bacterial biomass. We use RBC, we quantitate that as a surrogate of diatom and green algal abundance. And also we look at the biosynthetic genes that support TNO production, because not all organisms have the ability to make TNO. Now, what I would say is if you want a detailed run through of what goes on in the laboratory, what we're going to do is post a video, again, a nice video of me in the laboratory, which is a very rare event, taking you through that. And again, the team here will make sure that video is available. You can see how the various stages of sample processing are done. So what about functional analysis? Well, there are 2 biosynthetic pathways that support the production of MIB and geosmin, and they are separate pathways. There's an integrated biosynthetic secondary metabolism pathway that leads to the production of the precursors, and there are 2 critical enzymes that convert those precursors into these taste nodal compounds. Now, for 2-MIB, it's 2 enzymes. For geosmin, it's a single enzyme, and these enzymes are encoded on specific sets, or we call them operons, microbial operons. And those microbial operons actually are not in all cyanobacteria. So they also jump around. So this is a study that was done that looked at the phylogeny of the 16S against the phylogeny of the GO, the geosmin synthase gene. And basically what this shows in a very complex way is that actually the trees do not match. And that's because the geosmin is being horizontally transferred, so it can be acquired by individual organisms. It's not like passed down from father, passed down in sort of standard inheritance. It actually can be transferred across. And here's an example that sort of illustrates the problem we have. So these are 3 individual isolates of a particular cyanobacteria, Floss aqua, and the first 2, a full genome has been sequenced and documented, and you can see this gene REC-A. Now, REC-A is one of those genes that every bacteria has. And you can see that that's there in those first 2. What you can see in the third isolate is you can see the geosmin gene and the 2 auxiliary genes. And that's only in that one isolate. So even though these are identical species, one strain has the geosmin biosynthetic pathway, the other do not. And that's the issue. It's not just about identifying your species. There are specific strains that have and some that do not, and strains can basically acquire the ability. So as well as identifying the potential strains that could be involved, we also want to go down and see if they have the functional potential to make the geosmin. And we do that by looking for these biosynthetic pathways for both the MIB operon as well as the geosmin operon. And again, we can then use qPCR, a quantitative method, for quantitating how many organisms within a population, within an eDNA sample, have the ability to make geosmin and, on the other hand, MIB. So we quantitate the potential of those organisms to make the enzymes that produce the taste and odor compound. And actually, we have a good correlation. Again, we'll come back to that, from the copy number of those biosynthetic pathways to the production of geosmin. But it's not a perfect relationship, because just because they have the potential, if the nutrient environment is not correct, they're still not going to make those enzymes. So are there directions that we need to travel? So this project's been incredibly fruitful. We have a toolkit which is fit for purpose and which we can deploy, but where are the areas where we can improve things? Well, actually, I'm always talking about improvements. One of the improvements is this big idea of amplification. So the amplification, the meta barcoding that we do, that Sophie took you through, does have some technical issues. Again, it creates various issues with the data that can be improved on. And one of the improvements is to go for metagenomics. So we don't do an amplification, we just take all the DNA from an environmental sample and we sequence the whole lot. And if we sequence the whole lot, it basically provides us with a much more accurate representation of what's in the sample. Now, the thing is, that representation needs information to inform it. So we basically need a database to interpret it. And actually what I've done is I've pulled together a series of numbers that show you the expansion of the sequence database. So that's the interpretive power that we have over the last few years. And what you can see is if you look at the 16S cyanobacterial graph, again, look at the numbers. That's the number of sequences that have been deposited in the database for cyanobacteria. That's not individual species, that's accessions. And there's hundreds of thousands of sequences now representing cyanobacteria in the database. So if we have a 16S sequence, we can identify the species very straightforwardly. For diatoms, you can see we're in a better situation than we were. We're not in the same as cyanobacteria. So we have some thousands of information. So we have about 6,000 diatom sequences for RBC, which is the barcode for the diatoms. It needs to be included. Diatoms are incredibly diverse. We need more of those RBC sequences, but we're in a good situation. Now look back. When I started a project with the Environment Agency in 2010, between 2010 and 2014, doing diatom analysis, and you can see that in 2014 we barely had 1,000 sequences. So that database has expanded hugely, increasing our interpretive power. Now, if we're to do metagenome, we don't just need those barcodes, we need whole genomes. And if you look at the cyanobacterial genome, we actually have about 6,000 cyanobacterial genomes, full cyanobacterial genomes. So that means that we can actually do metagenomics on bacteria really effectively. Diatoms, we've barely got 34 diatoms, and there are hundreds and hundreds of diatoms that we need to characterize. So we're really hoping that projects like the Darwin Tree of Life project will come along and fill that gap. obviously, if it's very important to the industry that we understand the algal community via metagenomics, maybe, you know, we can fund some of that work as well. But the more knowledge we have, the more interpretive power we have. We've also been looking at nanopore and comparing it with Illumina. We've also been looking at barcoding versus metagenomics. And without doubt, the metagenomics gives us a more accurate representation of communities, but we are very keen to do this technical work to ensure that what we generate is supported by a good evidence base. So again, doing all of the standard, this is a mock community, a known community of microbes, and ensuring that our techniques are accurate and represent that community accurately. So again, as well as doing the discovery, we also want to do the QC. And finally, this is just a metagenomics study done by an American group showing that if you use metagenomics, you can find not only your offending organism, but your offending biosynthetic pathway. So again, identifying the organisms, but also the functional pathways within them. Now, Sophie did talk about nanopore, and I want to talk about a few future innovations. So one of those is the nanopore. Now, it's easy to say these things are small and efficient. I'm just going to pick one up. So here we go. This is a nanopore sequencer. You can see it's barely the size of my hand. The whole idea for thinking about this technology is to democratize genomics. So basically, we have this in universities, but why shouldn't you have this in, you know, for instance, at the end of a reservoir where you actually need the information? These are relatively easy to use. The reagents, although they need a fridge or a freezer to maintain them, can be taken out. The sample can be processed relatively effectively, and you could use this to generate sequence data, as it were, in situ. Definitely any of the laboratories supported, definitely the Welsh water laboratories, which are actually phenomenal, could definitely support a sequencer like this. And basically, they've got consumables. I've got some consumables here. This larger consumable item would allow you to analyze 96 samples, whilst this smaller one would allow you to analyze about 12 samples. And that's only costs about £80. So you can divide that. You need some additional reagents, so it's not quite under £10 a sample because you have to produce the samples. These actually have the compute power for a lot of the interpretation in them, and also they can be connected through to a computer, so you can see the samples, the data not on the machine, that's a touch screen, as well as on your local laptop. Now, you might have some people tell you that, oh, isn't that data very error prone? Again, it's a technology that was introduced, it's now quite old, but it was introduced years back, and at the time had a very large error rate on when it caught basis. And what I've done is I've supplied some information. There are always technological developments, and you can see that basically there's an algorithmic development that makes it sort of now sort of 98% accurate, so 98, 99% accurate. So again, this kind of development in the technologies and reagents are making this a very good leader for the future. And again, the size and the ability to have one that's very portable, I think, is a huge advantage. So what about making things more rapid in the future? So are there ways of speeding up? What does the research horizon? So we have something that we can deploy now, but what are the sort of questions that we want to ask in the future? Well, I've given 3 directions which I think are really important. The first one is people say, well, you've got DNA, what about RNA? So the difference is DNA is the template the organism uses. RNA are the genes that are actually used by the organism, that are transcribed and eventually made into proteins, which are the sort of mechanics of the cell. Now, actually, we can do RNA. The difficulty with doing RNA with the kind of setup that I've shown you is the filters would need preservative, because the RNA molecule is actually sensitive to degradation, much more sensitive than the DNA. So unfortunately, you can filter in the same way. That will give you the RNA, but you would need to preserve the filter on site. And as I said, that means providing a preservative to the person doing the sampling and training them how to use that preservative. And although the preservative is not highly toxic, it is the sort of thing where you need to basically have the health and safety training to use it, because if you were to get it in your eyes, it would not be a good outcome. So again, it means some complexity on the sampling and probably training for the samplers. And as I said, a number of industries that we have worked with basically are using independent sampling people who don't have the biological or chemical training to handle that kind of preservative. But it is a possibility. And we would love to do this sort of study, because what it would tell us is they have the biosynthetic gene, but are they making the biosynthetic gene? And if they're making the biosynthetic gene, we can give a good anticipation that that gene is then being used for the production. The other one is mesocosm studies. We have actually a part of a European consortium that are looking at mesocosms, where we can alter the nutrient compositions of the different mesocosms to try and investigate what actually stimulates the production. So if they have the potential, if they have the biosynthetic pathway, how good is that? But what nutrient combinations can cause the compounds to be made, not only made, but also released into the water. And again, we've done lots of studies to look at that. Rue is much more informed than I am, but to look at the nutrient triggers that can cause taste and odor production. Finally, obviously, we look into a crystal ball, and wouldn't it be nice to engineer an organism that perhaps gave us a direct indicator, maybe changed color, created a fluorescent protein, when a taste and odor event was possible or could be induced. So again, we could do that. It's one of those things that's very standard now, to genetically or engineer an organism that perhaps would change color when there was the potential for TNO production. But again, all of these are research areas where we could take the research further forward. Now, I'm going to hand back over after the break to think about the things we've learned. But the one thing we've learned more than anything else is that working with the industry is incredibly important, because it allows us to co-design the research we do to ensure that it's fit for purpose. Without Paul in this study, we would not have actually been able to do anything. Without Rue's first interaction with Welsh Water, we wouldn't have known the problem and how to develop solutions for the problem. But more recently, we've worked with Jenny at Yorkshire Water, Matt in UU, and Sam in Bristol Water to ensure that future projects is designed to actually address that industry need. So hopefully I've shown you a little bit of insight into the technologies. Sorry, the pumping didn't go as effectively as I would have liked it to, but hopefully you can see that this is a relatively simple process that can be used in the industry to give you not only taste and odor information, but address a lot of spectrum of issues that you guys are facing. So, Zoe, I'm a little bit early because I think we had till, but it's nearly 11 o'clock. What would you like to do? Great. So we're going to have a break. We're going to have a break for about 15 minutes. So come back at 10:15. Oh, 10 past. If we come back at 11:10, and then I think there's a little bit of additional presentations about what we've learned, and then there's an ability for you to do a Q&A and ask the team any questions you have. So I think Zoe's going to come in and do the technology side. And I look forward to speaking to you all after coffee.
Project since, and that should lead nicely into all the Q&A session. So do remember to add your questions to the chat. Again, so I'm just going to keep swooping towards. Oh no, I'm not. It won't move.
That's fun.
Bear with me 1 second, guys.
Use the hour.
A small technical issue with the clicker again. It's not playing ball. Just bear with. Should close it and open again? Yeah. Just bear with us, sorry, 1 second. I'm just going to do a quick test. There we go. Great. Okay, I'm just going to move this out of the way a bit so I'm not still on my magic cross. So you're probably very close to the screen. So I'm just going to quickly go through what we've learned. You can't. So yeah, good sample size is really, really important to give you a good resolution of data points around your reservoir. You get a lot of changes in community kind of between time points as well as spatially across those reservoirs. So collecting data on a regular basis is really important. So we started out kind of collecting samples every month. We've now done them every other week, but you can collect as many samples as you want within reason and within budget. So obviously there will be a kind of limit to that in terms of staffing time and budgetary requirements. But what you're looking for is that representation. But what is really important for us is that there's no missing data, especially if you're going to align the eDNA data with other types of variables that you're interested in. And this is important when you consider that when you are doing the bioinformatic process for this, you may lose a few samples along the way that don't meet a quality criteria. So if they are not high enough quality that we can confidently assume that the information is correct, they will be removed from your data set. So you have to bear that in mind as well. Missing data points are really, really important for us for so many reasons, but when you want to look at the data with any kind of confidence, ideally what we want to be able to do is model that data eventually. And what's important to understand, which some of you may not, and that's fine, but in statistical modelling, the programme assumes that you have an even data frame. And what that means is it really doesn't like NAs. But more importantly, a lot of people think that when you've got a lot of NAs in a data set, just those individual NAs are omitted. And that's not how it works. Because it needs an even data frame, the lines of data are deleted. So where there is absent data, there is an absent row. So if you can see this picture on the right-hand side, you go from 9 data points to 6, which doesn't sound too bad. Sorry, we're changing slides again. Which doesn't sound so bad, but actually this is a real example. This is a real data set that we were sent through. This wasn't sent to me. This was sent to a colleague. but I thought it really nicely explained this. So the colleague was sent a data frame with 1,839 data points in it. This is a real data set, and it had that many data points in it to begin with. And when looking at the number of variables that aligned, they ended up with only 13 data points at the end. And in one of those scenarios, they only ended up with 2. Now, there are certain ways around this, for example, removing columns of data, so particular variables that have a lot of NAs in them. But sometimes it's not easy to see quite how many of those NAs don't align, how much data you're losing. So going from 1,839 observations over a very long time frame, they ended up with 13 observations or 2 observations, which you can't model, and obviously you can't make confident predictions on. The other thing that's been important for this project has been really regular meetings. So as an absolute minimum, we've been working with monthly meetings where we get all the partners together. We have a big discussion. We talk about what we think is working, what we think isn't working. But actually, even more regular meetings would be even better. The more regular, the better, especially while you're setting it up. It's important for both directions. One, that we are kind of liaising with them to be able to make sure that we're getting the data feedback that we need, but also so that we can make sure that we're making improvements that are benefiting the collaborators. Another thing which I guess a lot of people maybe don't consider, but we have had a couple of times throughout the project where people have left the company, and there hasn't been necessarily the easiest of handovers, or inconsistent staff working on the project, which means that either there's been a delay in how often we have to kind of re-explain everything again, which is fine. We don't mind communicating this information, but it has led to a lot of delays downstream where we then ended up missing data points and sampling points, which is a difficulty. Another small one that's worth considering is so not everyone does their own sampling. There are lots of options for that. So if you don't have the staff in time to conduct your own sampling, you can employ other companies to be able to do this. There have been a couple of scenarios where we've done some of the sampling, which does take away some of the time that we have in the lab and the staffing times around that. But the main thing is we don't want to be sat on this information and not getting it out there, because we're all working as a community on this problem. The knowledge exchange and the technology transfer is just so important. So we've worked at loads of different events, including this one where we've kind of talked about the work, but we've also had people here in Cardiff University where we've invited people in, people who've even requested it. That's totally fine to request training sessions if there's enough of you, where we've then kind of explained the eDNA in depth, explained data in depth in some instances. We've shown people how to analyze that data, but we've also done work helping them to understand how Tableau works, how they can get their own data up there, but also maybe also showing them how to understand in an easy way what their data looks like, which is really important, again, for making sure that we've got that handover where we're then amending what we need to. So now what I'm going to do is I'm just going to hand you over to Rupert Perkins again, and Ru's going to kind of talk about where we've gone with this project since.
And thank you, Sophie. And boy, was I glad I wasn't the first one on after the break. So I've been handed a new clicker. I'm hoping it's going to work. I'm not quite sure which buttons to press, but we'll find out in a minute. Okie dokie. So the way forward. So what we've been talking about so far is basically the original Ofwat project that was ultimately finding out what industry needs were, trying to find out a technology system that we could actually put in place to help with finding solutions. What we now need to do is move forward and demonstrate this is actually working and is a useful tool within industry to give further evidence of why we think water industry should be investing in this as a way forward to augment existing aspects of catchment and reservoir monitoring. So we've got one initial project that I wanted to talk about, which is our When and How to Mix project. So this is an industry-funded project with a whole bunch of water companies collectively funding this, and those companies, thank you very much indeed. You're named there in the top right-hand picture of this slide. So what we're talking about now is taking eDNA into holistic reservoir management. So if you are just focusing on taste and odour from geosmin and MIB, from cyanobacteria in a reservoir, you might be thinking to yourself, okay, I'm addressing that problem. But what happens if your intervention solution makes a different problem worse? So the picture there is of a ResMix turbine, which many of you will obviously know about. But what about if it's a bubble plume or a bubble curtain or any kind of forced mixing tool? So if you're in a reservoir and you're trying to prevent stratification, or you're trying to induce mixing to break down internal loading of things like manganese, you're often using these types of forced mixing systems for reservoir recovery. Manganese, as we know, comes from the sediment as a soluble form, as well as other sources. And we also know that nutrients come from the sediment, such as ammonia and phosphate. So if we're looking at taste and odour, and we're looking at cyanobacterial producing metabolites like geosmin and MIB, we know from studies like our previous publications that spikes in nutrients can lead to these taste and odour events. But at the same time, spikes in manganese can occur at the same time as spikes in nutrients. But it's not as simple as that. If you put a ResMix turbine into a reservoir and you start mixing it, or you put bubble plumes in, or whatever your kind of mixing technology is, you can try, if you get it perfectly right, to prevent the internal loading of manganese and of nutrients, but you're mixing up the water column. Now, our cyanobacteria, from a biological point of view, are absolutely fascinating because they're amazingly well adapted. And they will actually be able to utilize the nutrients in deeper water. And also, they like lower light environments. So if you start mixing your water column to prevent stratification, to reduce internal loading of manganese or or nutrients, you can actually increase your taste and odour risk by taking the cyanobacteria from the surface waters, mixing them down so that they get the nutrients, bring them back up so they get the light, and they get pulses of nutrients and light. Now, we know from our previous studies that it is pulses of environmental conditions and pulses of nutrients that creates the perfect storm for taste and odour events. So as a result, what we are saying is, if you're doing any kind of management within a reservoir, think holistically. Think about the whole bigger picture. Don't make one problem worse by treating a different problem. So within this project, then, we're using eDNA, but we're using it to address many different aspects. So we're focusing on mixed sites and non-mixed sites with a range of water companies. And we're looking at how basically this could be controlling manganese. So we have colleagues at Bath University, led by Lee Bryant, who are working on that. And we're working in far more detail on the algae and the cyanobacteria and the community linked to different types of water quality risks. So that could be, for example, algal blooms, which is a distinctly different water quality problem compared to our taste and odour that we've been focusing on today. But we're also looking at the organics that are produced by these algae that are linked, for example, with disinfecting byproducts, DPPs, produced in the water treatment works. So we're using the eDNA community integrated with mixing, integrated with environmental conditions, integrated with all the other variables we're measuring in sites like nitrate, ammonia, phosphate, etc., to try and find out what is the combined water quality risk levels within the reservoir. Because one thing we want to emphasize is that eDNA isn't just for TNO and for cyanobacteria. It depends on the primers you produced and how you approach this with the pipeline to actually analyze it, but you can use the same samples for a whole range of different problems. So we're integrating the community of algae that we're studying with eDNA to try and link that with a range of different problems, and also with non-biologically associated risks such as manganese discoloration. So that's one of our key projects that we're doing at the moment. And I want to just quickly mention, because some of the questions that came in during the break, Within this project, we've got a lot of value-added projects. So, for example, I gave thanks to colleagues at the beginning, such as Annalise Hooper. Annalise's project has been a case-funded PhD with Welsh Water, and Annalise has been looking at the different triggers for actually not the taste and odour metabolite production, but the elevation of the genes that Pete was talking about that are producing the metabolites. So what we've been doing is looking at the mechanistic pathway that has enabled us to actually link the eDNA community with the actual species that are producers, identifying specifically which species we're talking about in the reservoir, then the gene copy level that they've got for the genes that they're making the taste and odour metabolites, and then correlating that with those metabolites so we know that that gene elevation is actually linked to the actual taste and odour event. So it gives us the mechanistic story. And then from that, we can take that in understanding of the data we've got for that, the quality of and the magnitude of those relationships, to embed those into this project and give us far more learning that ultimately generates knowledge for evidence-based management, but also a predictive capacity for determining when taste and odour events will actually happen. Trying the clicker now. Okay, the next project then is basically almost sort of tying the loop, if you like. So we started with eDNA. We started with the eDNA at the sort of technology transfer approach. What we're now doing is another Ofwat funded project in collaboration with Bristol University. And Bristol University worked with Welsh Water looking at using artificial intelligence for trying to create machine learning tools to take microscopy, but take the difficulties that I referred to earlier on in identifying species and making a computer do it, basically. Now, we've got a bit of a loop there, a bit of a sort of a tautology, if you like, but ultimately, if we take artificial intelligence and computer machine learning to identify species, that's only as good as the data it's been given to tell it what those species were in the first place. So we need the eDNA to ultimately ground truth that so that we know we haven't got observer bias or misidentifications, and also to take it to the next level. Can we get the computer to identify things that that somebody in a lab can't identify themselves? So can the computer see things that we are not seeing, so that we can actually then relate this not just to the species, but to the functions and the problems regarding the water quality risks. So this is our current project. We're embedding the eDNA, but at the same time advancing our community data analysis, linking all these projects together for more value added and synergy that enables us to really use the biology as the indicator tools of the water quality risks. So that's it for what we're doing at the moment, where we're carrying on with this original project. And we're going to be moving over now to the question and answer session.
Okay, so hello again, as if by magic. I'm on a different microphone. I don't have a clicker to contend with, and we've got our questions coming in. So I'm going to pass to Pete, who's got the first questions written down. Over to you, Pete.
So what I've tried to do is paraphrase your questions and group them together so we can do a sort of bit of a logical walkthrough. So the first one, I suppose, is I think the most relevant to this group, which is how do the sort of eDNA analysis we've done relate to the sort of metabolic levels of TNO concentrations and compounds? Roo, do you want to start off with that?
Okay, so I sort of started to address this when I was talking about Annalise's project. So what we've been doing is looking at the eDNA community, and we've been looking at the copy levels of the genes responsible for producing the metabolites, and we've been looking at the metabolites themselves, and we've been analysing all these data in connection with the metadata in the water quality data provided by the water companies. So we're putting that all into a bigger picture, if you like, modelling approach to actually make sure that what we're measuring is mechanistic. So The elevated copy levels, for example, by the cyanobacteria, we know that correlates very closely with the geosmin levels in the reservoir. We're also able to analyze that and start to look into the lag times between the changes in nutrient parameters, for example, which we've been working on for some years now. So we know ammonia to nitrate ratio is a critical trigger, and we're able to look at the metadata to see when that trigger actually occurs. We're then able to look at the lag time in the production of the elevation of the copy level, and then the production of the geosmin or MIB. Geosmin works better than MIB in the actual reservoir. So that's a preliminary answer. Pete, do you want to add anything to that?
The only thing I would say is that obviously complex, the advantage of what we're doing is building up that knowledge base and a modeling interpretive base, so that actually what we hope is that if we get the information soon enough and fast enough, we can make accurate predictions. Making all the measurements is one thing, but having the interpretive power is another. So again, we can do speciation by eDNA, but in the end, you have to put that together in a holistic manner to interpret what's going on with your reservoirs. So the next question actually is a really good question. It's about the persistence of eDNA and whether actually, because eDNA persists for quite a long time, whether we're measuring something that happened in the past or whether we know exactly what's happening. So again, I'll hand back to Rupert for an initial answer, but I think this is technology, so this will be thrown back at me. But really, do you want to give us a...
If I can throw bits back at you, Pete, that'd be good. Okay, so Pete knows far more about the eDNA, but let's approach this question purely from the point of view of water industry. So the water industry want a tool. They don't necessarily want to have the integrated step-by-step stuff that I've just been talking about. They want to know, have I got taste and odor looming on the horizon? Is that the issue? So ultimately, what we're doing with these methods are demonstrating how you can integrate measurements of eDNA over time to generate community patterns over time. So my analogy would be, if you have ever worked in stream ecology or anything like that, you'll know a process called the biological monitoring, BMWP, Biological Monitoring Working Party. Now, if you go out to a stream and you take a kick sample, you can collect the invertebrates. The invertebrates are there over time. They are integrating what's happened in the water chemistry over time and the environmental variables and the whole multivariate triggers of what happens within a stream. They are therefore much better at telling you the water quality than going out and collecting a bottle of water and measuring it on the day. So ultimately, I would turn this question around and saying the fact that eDNA persists gives us the ability to look at the community that is persisting over time and the changes in that community, and using that duration and changing community to be the tool that is our biological indicator of risk. So ultimately, we'd be stuffed if the eDNA didn't have any kind of persistence, because we wouldn't be able to see the persistence of the community, and we wouldn't be able to obviously go out and sample at a sensible time period, because ultimately, if eDNA didn't persist, we'd have to be going out there every single day. As it is, we see going out about every 2 weeks gives us a really good data set that enables us to link that with the actual water quality risk.
And of course, the 2 weeks is not a random number. So again, there are some nice publications in the public arena. The first one by a guy called Eske Villeslav from Denmark, where they actually looked for the length of eDNA persistence in water bodies, and different water bodies, of course, different persistence, but in lakes, it's about 2 weeks. So again, you can see that our number of 2 weeks for our intermediate sampling is not a random selection. The other thing is, if any of you are more interested in this, there's a really a great eDNA expert called Cy Creer from Bangor, and he's worked with Isabel Duance here at Cardiff University to do various timings. So what they did is they introduced a foreign DNA sample, which actually was a marine fish. Obviously, when you're looking at freshwater systems, they don't usually have marine fish in, and they basically looked at the persistence within those waterways. And again, they did look at lotic, so basically flowing water. And again, if you have the hydrological characteristics, the temperature, the flow rates, you can actually predict how far eDNA goes. So again, Cy Creer, Isabel Duance, and a project called LowFresh will give you that technical information if you're interested. So The next question was about horizontal gene transfer. So I touched on this as far as the acquisition of the production of MIB and geosmin. You're happy if I take it directly?
Go ahead.
So basically, we know that there are lots of acquired characteristics, phenotypes, and the ones that we know a lot about, of course, are antimicrobial resistance, the ability for microbes to acquire a function. And we do think that MIB and geosmin can be acquired. As I showed in my presentation, we have species where we have some strains that have the biosynthetic pathway and some that don't. Of course, it could be lost, but actually it does look like it's being gained from and transferred between organisms. Now, there's lots of technical ways you can do that, transposons, plasmids, etc. We do not see it on a mobile element. So it's always integrated in the genome, and therefore it's not as easy to transfer. But we can see no signatures. Again, there are particular genetic signatures that facilitate things to transfer. We don't see those. So we don't know the mechanism of transfer. We don't also know the selective advantage, because if you have a gene like the productive gene that makes geosmin MIB, for a microbe to keep it, it has to have some advantage. So it must be giving the organism some competitive advantage to be kept by the organism. We do not know that. We have hypotheses. Rue and me would quite happily spend all day telling you why we think they need MIB and geosmin. And that actually comes maybe related to the next question. But again, that's a kind of research area that we would love to explore, both looking at horizontal gene transfer as far as mechanistically, but also the drivers of mechanism. And there are also other relationships. So we talk about networks with diatoms and other algal species, and we know that the algal species and the cyanobacterial species, so basically the diatoms and the cyanobacteria, also interact in mutual networks. And again, there are key things. So again, if you're interested in us investigating those research questions, we'd be very happy to do so. But again, I'm think as far as giving you a tool that actually delivers for your everyday, those things are future rather than present. The connected question that I'm going to throw at Rue at the same time as handing the microphone is, we're seeing more after climate change. Could climate change or any other change be driving the increased frequency?
So I'm going to link this to two associated questions, because I noticed another one, which was talking about whether the horizontal gene flow could be effectively one of the causes of why we're seeing taste and odor in our upland oligotrophic sites. Connected with that, I would say probably not. What we're seeing in our upland oligotrophic sites is the classic model that we've been explaining in our publications and reports to industry, which is ultimately what drives taste and odor metabolite production isn't basically, say, something like oligotrophic compared to eutrophic. It's the change in nutrients, and when those changes in nutrients interact with environmental conditions to give you the perfect storm. So if you're in an oligotrophic upland system, or even a mesotrophic upland system, ultimately you have lower background levels of nutrients, which means if you get a change in nutrients, even if it's a small change, the relative magnitude of that change is then higher. And as a result, that can be what changes, A, the community that is giving you the taste and odor risk, but also the metabolism of the taste and odor producers within that community. So in our upland sites, it's a perfect example of where a small change in nutrients becomes really important. So size does matter. I'd link that then to the question about climate change. And undoubtedly, in my opinion, climate change is increasing not just taste and odor, but many of our biological algal associated water quality risks. Obviously, if we're looking at the classic drier summer periods, those drier summer periods are particularly when we get shallower, warmer reservoirs, which favor cyanobacteria. They also favor different types of nutrient releases that ultimately can cause these small changes in nutrients that can cause our related taste and odor risk. But also we're seeing pulses of rainfall in summer, which are then linked to changes in nutrients as well. So Helen Cantwell, who did a PhD with us, was actually able to demonstrate that if you get a pulsed rainfall event, or you have a pulsed period of refilling your reservoir, ultimately those two instances can actually decrease the amount of nitrate concentration, but increase the amount of ammonia concentration. So increasing your ammonium to nitrate ratio. And we know ammonium to nitrate ratio, when that goes up, it increases your taste and odor risk. So not only are we producing a warmer, drier summer that favors cyanobacteria, but the periodicity of pulse rainfall events and the more sporadic weather patterns we're seeing are classic drivers of the perfect storm for a taste and odor event.
Thanks, Ru. I hope that covers that question. Now, there's a whole lot of integrated questions because many of you have been taken with the suggestion that Sophie made about other applications, how you can use the technology. And I'm going to lead off with one that's technical, which is saying, how do we tell the difference between bacteria and algae? And then people are asking about crypto, obviously always crypto, Actinomyces, pathogens, AMR, and fungal. And again, I'm going to take all of these together bizarrely, because what I'm going to do is reiterate what Sophie was saying. So our eDNA sample is like a library, and the library contains lots of information that can inform you about a whole suite of challenges that you face. When we take a particular set of primers, we target a part of that library that tells us about a particular community. So if we take primers designed towards the 16S ribosomal RNA, those are designed to amplify a gene that's very well characterized in bacterial communities. So basically, we're taking out the bit of the library that's on bacteria and looking through the species of bacteria that are there. And we can look at any bacteria that's there. So if we're looking at fecal coliforms, we can look at those E. coli and those other bacteria that are associated with coliforms. If we look at a different marker, and again, the one we use for algal is RBC. So that's a rubisco gene that's, of course, associated with the harvesting of light. And we design primers, and in fact, we had to change because initially we designed primers for the Environment Agency that are based on diatoms only. And again, if you want to do water quality analysis using the Environment Agency prescribed method, basically you want to use benthic diatoms. And again, we can do that, but that's separate from these. These are actually the organisms that live in the water body themselves, not the benthic ones. And again, we have redesigned those primers so they allow us to look at diatoms as well as green algae. And therefore, we can get the full community and the interaction between those two communities. Now, let's say we didn't want to do algae, but we wanted to do fungi. We would use a different set of primers and allow us to access a different part of that library. So we would look at what's called the ITS region. You hear it's a very sort of molecular biology, word that everyone throws around. It's basically a spacer region between two ribosomals, and that's used for fungal community analysis. So by choosing the different elements of the library we want to access, we can address different questions. Now, crypto is really high on everyone's agenda, and obviously there's a legislative process. I would say, again, there are some partners that are in the room that have done some fantastic research on developing crypto-specific assaying. Now, there are two. I'm not going to say anything more because it's up to them to share when they're ready to share. But I think they've had a lot of success in genetically profiling those crypto communities and getting a method, because actually we capture in the eDNA, we would capture cryptocysts, but cryptocysts are incredibly resilient to being lysed. And although we think our method does, it would need to be validated. The other thing is that when you're looking for crypto, you actually have a prescribed frequency you need to look at. So today we usually sample about 500 mils of liquid. If you wanted to look at crypto, you'd need to be sampling more like 10 liters. And although the filters will do it, it'll take a lot more time. And in fact, there are prescribed filters with much larger density that can be used. Now, can we take those larger filters and do molecular biology to characterize crypto? Yes, we can, but there is a slightly different method and it would need to be refined. As I said, I know there are partners that are probably in the room that have that information, but it's up to them to share when they're ready. So hopefully that shows you there's a whole suite of things that we can do. I would flag fish stocks, relatively straightforward. Invasive species, relatively straightforward. And again, protected species, relatively straightforward. Again, there are some come across them, and their managing director, Kate Bruce, she is absolutely fantastic. And again, they specialize in those kind of nature-based analytics. Rue, would you like to add anything?
Just two quick things to add to that. The first thing comes back to what Sophie was saying earlier on, which was she mentioned in passing nature-based solutions. One of the key things I think going forward is that what we're talking about is taking one sample and being able to run that for a whole range of different types of organism, whether those are your aquatic macrophytes, your algae, your cyanobacteria, your bacteria, your fungi, your fish, birds, you name it. That gives you then the full freshwater community in your reservoir. Ultimately, if you want to start investigating into nature-based solutions, you need to know what's the capacity for manipulation to actually facilitate that nature-based solution. So therefore, understanding what's there and why it's there is the crucial first step in that process. So having this suite of different aspects of information about your system enables you to know what's the capacity for working with nature to try and find a solution. And I think that has to be the way forward for water industry, really, to actually think about how biology can be used as an advantage to maintain clean water and reduce costs of ultimately treatment and also reservoir management. That's me on a soapbox. I've got one other quick thing to say, which is one of the questions in the chat was talking about bacteria compared to algae. And so sort of linked to that is, why are we studying this full community of bacteria and diatoms and green algae and cyanobacteria in our modeling approach? One of the key things I wanted to say is that we know from looking at the species we're getting that we can understand why we see associations between different groups because of certain biological interactions. So, for example, we know that there are certain diatoms that associate with cyanobacteria because the bacteria are cyanobacteria basically stimulating the diatoms to actively produce nutrients for them. There are certain bacteria which have feedback loops with cyanobacteria which enable ammonium levels to be increased. So we know a lot about the different interactions between the groups, and that gives us far more of a capacity for basically understanding the problem, but also using the biology to indicate what's happening in the reservoir. And that therefore needs to go to the next level. So when Pete's talking about looking at invasive species, do the invasive species put that balance, that ecosystem, that community out of balance? Do we need to look at the level of zooplankton in connection with the algae? Do we need to look at the fish who are feeding upon the zooplankton? We need to understand all these interactions, and the eDNA is most definitely a really good quantitative tool to actually start looking at the interactions within the community.
Thanks, Rue. A couple of others. I'm going to switch to a practical one and throw this at Sophie. So some people are asking about turnaround times, Sophie, and what's the turnaround time for processing a sample?
Yeah, so the turnaround time varies depending on the technology that you're using, but for both of them, they're relatively quick. So within our team, we have Charlotte Taylor, who is our technician, our molecular technician, and she is the only molecular technician that we have working on this. And she is able to turn around on top of a suite of other things. The data for me to be able to get it online every 2 weeks. And by that, I mean, extracting the DNA. So there's some pre-prep work, extracting the DNA, taking it through that whole methodology that I showed you earlier. But alongside that, she's also able to run qPCR and stay on top of other laboratory things. So it depends on how many samples you have coming through and how you'd like to approach it. What we've been doing is a combination. So we do use nanopore a lot, but if we are running lots and lots of samples, we've created a plate approach where essentially we can split the plate into 3. So it's a 96 well plate. So depending on the number of samples, we can do 1/3 of a plate, 2/3 of a plate, or a whole plate. And that's really enabled us to run a lot of samples quite seamlessly. There's other things that we can utilize within the university to speed it up if there was a bottleneck. So there are certain roboticized tools that we can use for basically just automating different sections of the protocol, and they can be really, really useful for helping take some of the pressure off if needs be. They are relatively expensive, and you can do without them. It does basically what the human does, but a lot
quicker. So if you kind of hit a bottleneck, they can help. But yeah, so we aim to get the data up, analysed and on the interactive Tableau dashboards every 2 weeks.
Sorry, Pete, just quickly, some people listening there might be saying 2 weeks. That's telling us what happened 2 weeks ago. What we do with the data ultimately, which comes off the back of the Bristol project that I mentioned earlier on, is we are basically trying to predict what happens in 4 weeks' time. So ultimately, 2 weeks is when we get the data. So that's looking back 2 weeks. So we predict what happened from 2 weeks ago in 4 weeks' time. So you've got a 2 weeks prediction period of what will happen.
And that nicely allows me to go into the second part of the question, which is about innovations. So if you do have that 2-week warning, what innovations can you make on the back of this to prevent TNO complaints? I'm going to hand to the expert here and hand to Paul.
Thanks, Pete. I think there's 2 things we can do. We can look at catchment interventions, but they're more long term. So if you understand what's going on in the catchment, you can try and prevent the nutrients going in or whatever. And then obviously on a treatment works, it gets you ready to do things like blending, if you can blend with other sources, or activated carbon treatment, which will then take out your MIB and Josmin. Both of we really want to stop it in the catchment, but that's more long term. So short term, it would be doing something to absorb the MIB or Josmin, which would be carbon absorption.
I would also plug another project that Paul is supporting, which is looking at the biodegradation of geosmin and MIB within the treatment plants. Again, it's not for today, but again, Welsh Water have funded a project at Cardiff with a colleague of mine who's looking at basically whether there are specific microbes that are in the sand filters that can be encouraged to grow to facilitate biodeg within the treatment plants. But again, that's perhaps another showcase event that we could do. So finally, there's another question for Paul, which is, how do Welsh Water feel about the delivery of the eDNA projects, and what are the main challenges you experience from the water company side? It's very hard to ask this in public since we're sitting here, but Paul, be honest with us, please.
Yeah, I think because we've got the trust and we've built up trust over time, that's essential for any project that you're doing. So if you trust the people who are doing the project, then that helps. Also, if you have those communication lines open as well, it really helps. So to have those honest and straightforward feedback times for everything. And I'm trying to remember the second half of the question.
What problems? What challenges?
What challenges? I guess logistics are always the major challenge. How do you get the samples in a timely manner down to Cardiff? How do Cardiff then analyse them in a timely manner? And that's part of what the project was. It was producing all the standard operating procedures so we could get much slicker as we've moved on into our other projects, such as when and how to mix.
Again, that's a lovely name check. So I was going to mention when and how to mix. So basically, it's a new project which is directly funded by a handful of water industries that were keen to see the benefits of the system we've put with the different interventions that they're trying to put in tune with the reservoir. I'm going to say a little bit. I think I'm going to hand to Rue now, because there is a question that says, how is mixing different artificial mixing techniques affecting res mix, bubble plumes and bubble curtains? So I'm going to hand on to Rue to say a little bit about that.
So I can see that question comes from somebody who mixing is very close to their heart. So thank you for the question. That question basically is referring to obviously different types of mixing, which is really important. So what we've got within the When and How to Mix project are a series of sites that are mixed in different ways, as well as sites which aren't mixed. So we've got partners like Scottish Water, Jersey Water. So we've got a very wide sort of variation in climate and location, and in also reservoir type. So we've got a lot of different variables, and we've got different types of mixing within those different reservoir systems. So we're actually looking at bubble plumes, we're looking at mixing sites as well. So we're looking at the different types of mixing, and we're taking, well, the water company partners are taking water profiles, so we're able to actually look at the extent of mixing by different methods, and then look at for example, the changes in the manganese soluble and particulate, as well as the different aspects I mentioned earlier on. So, for example, things like dissolved organic carbon, the linked to the DPPs, the community structure that could be linked to algal blooms, and obviously taste and odour, which we've been talking about for some time. So from that question, yeah, we're looking at whole different types of mixing, but also within different reservoir types, because as you all know, reservoirs all behave differently, and therefore we need a suite of variation to actually try and find what's common between sites and what's different between sites, what's common between different mixing types and what's different.
Thanks, Ru. There's a couple of questions. One thing I want to just say is that on one of my slides, I mentioned mesocosms, and I showed a beautiful set of mesocosms. There are actually mesocosms at CEH. CEH have probably the best mesocosm setup that I know of in the country, allowing you not to do microcosms. Most people do microcosms. These are major mesocosms, and we've worked closely with them when we wanted controlled mesocosm. So anyone thinking about mesocosm studies, really, what's the lady's name? I always mispronounce it.
If I can just quickly add, yeah, Hydron at CEH. If you're online, hi Hydron. Hydron set up at CEH. That's within a large European project that Hydron leads. So we're just one of the partners there. But that's a perfect example of the connectedness between the different aspects that eDNA can be looking at. So partners all across Europe have been basically looking at, say, the zooplankton, the viruses, the algae like we are. So all sorts of different aspects of that project within those mesocosms. And obviously what we've been able to do in those mesocosms is actually spike them with different nutrients to see what happens to the community in response to that.
Thanks, Ru. Now, the last question, I'm just going to ask Ru to say a brief thing on AI. So basically, we have a new project you mentioned about AI. Of course, these days, everyone thinks of AI and maybe not in the most beneficial ways. So Ru, perhaps you'll explain, this is the last question about AI and how we're using it.
Okay, we haven't got very long left, and that is a very big question. So we're working with, it's actually Chris Williamson at Bristol University, who is an expert on AI and the modelling approaches that we can do with the data that come out of this. But basically what this project is doing is, if you look down a microscope, obviously you're identifying species. Now, Chris and Welsh Water have been working together I'm speaking for Chris, who's obviously not on this the projects we've been talking about, but Chris basically has been working closely to actually demonstrate that his techniques are able to identify species to a certain level, and that the computer basically can be used to drive the learning process to identify the species. So it's machine learning for identification of algae. What we're then doing is ultimately taking that to the next level, in that so far that project has been limited to ground truthing the learnt identification against observers in the lab. And as I explained earlier on, that doesn't actually tie up with identifying to the level we need if we're linking it to water quality. So ultimately what we're doing is then using eDNA to ground truth the AI learnt species identification, but seeing if we can actually go further than the microscopy in a lab environment to actually identify species, and potentially even linking that to our water quality risks.
Okay.
Yeah, so it's time to wrap up. Thank you ever so much for spending time with us today. I hope you found that interesting, and just getting a glimpse of what we can do with eDNA and how we'll take it forward in the future. So thanks for all your questions as well. Thanks to the panelists, especially the two Ronnies in the middle, and we'll leave it there. Oh yes, the poll. Sorry. So there's a poll coming up on your screen. If you could take time to answer it, that would be really useful. Thank you.
Thank you, guys.